Modification. A scientist sought out to discover the effects that a certain protein had on a eukaryotic cell. After some research, she discovered that the protein was the result of translation of DNA sequence located on gene A. She wanted to see what would happen to the cell if little to none of this protein could be produced. She searched for the cell that had a mutation on gene A so that the translation could not occur properly. However, after days of searching, she was unable to find one. Her research had come to an abrupt halt until a colleague recommended she try altering the genes herself. This way, she could have the right type of mutation that would benefit her research. She asked around about the best way to alter genes. Her colleagues gave her two answers. They had used both RNAi and CRISPR. However, she was unsure which of the two methods would work best for her research. She first started to look into what RNAi is. RNAi stands for RNA interference. RNAi is a method where RNA molecules inhibit gene expression by breaking up mRNA before it can be translated into proteins. RNA interference is a very simple procedure for scientists because it uses the cell's natural machinery to achieve the breaking down of mRNA. She continued her research by looking into how RNAi occurs inside the cell. First, dsRNA, or double-stranded RNA, is designed to attack certain parts of the mRNA sequence. Once inside the cell, it binds the protein dicer. Dicer cleaves the dsRNA into siRNA. The siRNA are loaded into the RISC complex. This complex binds to and cleaves the mRNA so that it is unable to be transcribed. If the mRNA is cleaved imperfectly, then some mRNA can be transcribed, but just in lesser amounts. Afterwards, she began her research on CRISPR. CRISPR's DNA sequence and associated protein Cas9 edits base pairs of DNA. Originally found in prokaryotes, it changes base pairs so that transcription cannot occur or that the sequence will be translated incorrectly. Again, she looked into how CRISPR works inside the cell. sgRNA, or single guide RNA, guides Cas9 to the target sequence in the DNA. If the base pairs in the Cas9 correlate with the base pairs in the DNA, the Cas9 will bind to the DNA. Once bound, it causes a double-stranded break in the DNA, leading to a deletion or insertion frame shift. After looking over her research, the scientist decided to use RNAi to modify gene A. She chose it for three major reasons. Firstly, it's an easier experiment to set up and much less labor-intensive to use. Secondly, it's able to attack a wider range of mRNA sequences than CRISPR can. And lastly, RNAi can both turn off the gene and reduce expression of the gene, while CRISPR can only turn off the gene entirely. I hope you learned a lot. Thanks for watching.